Bismillahirrahmanirrahim. Assalamu alaikum. The topic of today's discussion is about the cell cycle and cell. And what is cell cycle? Cell cycle is an ordered sequence of events that extend from a time when a cells divide from a parent cells until the time when the cells divide into two daughter cells. As you can see in the diagram that cell cycle is further divided into two phases. The first one is the enter phase and the second one is the M phase. During the interphase is also known as the growing phase, and it is further divided into G1 phase, S phase and G2 phase, and M phase is further divided into mitotic phase or cytokinesis phase. And during interphase, the most of the cell cycle spent in this phase, and during this phase cell metabolic activity is very high and cell performing various function inside the organisms, like intestine cells start releasing the digestive enzymes to digest the food. During interphase, cell make more cytoplasm and cytoplasmic contents divide, like different organelles, endoplasmic reticulum, mitochondria. These organelles get doubled and duplicate genetic material. Genetic material also duplicated in this phase. The second one is M phase. M phase is the division phase, and M phase is further divided into mitotic phase and cytokinesis phase. Now we will discuss the subphases of the interphase. The subphases of the interphase, the first subphase of interphase is the G1 phase, or you can say gap 1. During G1 phase, all the subphases of the interphase, during all the subphases cell grows in size, and during G1 phase protein synthesis increases. Protein synthesis increases because the proteins will need in next phases for different processes, like in the G1 phase. After G1 phase, S phase will follow the G1 phase, and during S phase DNA replication will take place. And during the DNA replication, different enzymes are required that are synthesized during the G1 phase. And in the lower eukaryotes, in some plants, histone proteins are also synthesized here. And RNA synthesis also take place in this phase. ATP synthesis also goes up, and raw material that is required for DNA replication is also synthesized here. Raw material like nucleotides that is also required during the DNA replication is also synthesized in the G1 phase. And in this phase, as I already told you, that in this phase cells increases. The second phase is the S phase, our synthesis phase. And why we called it S phase? Because in this phase synthesis of the DNA takes place. That's why we called it S phase. And as I already told you that during the S phase DNA replication takes place. DNA get replicated or duplicated. Every copy of a chromosome, every chromosome has a single copy, and during the S phase the single copy of a chromosome get doubled. And centrioles also divide. As you know that there is one centriole is present inside the animal cell, and during when the cell has to be divided, centriole is also divided, and it moves towards the opposite poles. And histone proteins are also synthesized here. As I told you that some histone proteins are synthesized in a G1 phase, and in some organisms like higher eukaryotes, all the histone protein synthesis takes place during the S phase. But in lower eukaryotes, histone protein synthesis take place in G1 phase. And as I already told you that at the beginning of the S phase, the chromosome was single. At the end of this phase, the chromosome become double, and each chromosome has two sister chromatids. If we talk about the start of this phase, there is 2 in number of chromosome, but every chromosome has single chromatid. But at the end of this phase, every chromosome has two sister chromatids. Or you can say DNA content present in one set of chromosome represent a NRC value, and unit is picometer. And after the S phase, 2NRC value is 4C. When we talk about the one chromosome, we call it as C value. And when the 2 in number, the value was 2C. And after the duplication or after the replication of the DNA, this value will be 4C. The third phase of the interphase is the G2 phase. And in G2 phase, here the protein synthesis also take place, because some proteins are required in the M phase, like tubulin protein that is required for the formation of the spindle fiber is synthesized in the G2 phase. ATP synthesis also take place in this phase. RNA synthesis. The next phase is the G0 phase, or inactive or quiescent phase. And some cells, after getting a normal size, they become inactive. After getting the normal size, they become inactive, and they remain at this phase throughout the life, and shall perform all the activities except division. They are performing all the activities. And if we talk about the neuron cells and muscle cells, neuron cells and muscle cells go into the G0 phase, although they are performing their functions, but they are not divided.